We can see it's only 5 to 8 micrometers long that convert the sugarish extract into beer. Similar yeasts make wine and bread and many other products for us. Laboratory strains, such as the ones we are now seeing, play a central role in today's biological research. When placed on an appropriate growth medium, yeast cells divide by budding. A protuberance appears on the surface of the cell and grows into the spherical bud. The bud neck is pinched off. Both the old and the new cells continue budding. In real life, the cell cycle is not a matter of seconds. It takes about 2 hours. A bud starts its separate life in stage G1. Each cell has a nucleus invisible without special stains. DNA synthesis begins at the time of bud initiation or a little before it. The S period of DNA synthesis extends over 1/3 to 1/2 of the cell cycle. The nucleus stretches out through the neck into the bud, then divides into 2 equal nuclei, one on each side of the neck. The cell cycle is completed with the formation of a wall between mother and daughter cells. The mother and daughter cells and all their descendants are genetically identical and constitute a clone. The yeast cell wall is largely made of glucan and mannan. The scar tissue formed on the mother cell after budding contains another polymer, chitin. The scar tissue is unable to bud again. Each new bud appears on a different spot of the mother's cell surface. After the mother cell has produced a score of daughters, it cannot bud anymore and dies. Yeast is thus among the simplest organisms whose cells cannot divide indefinitely but have a limited lifespan. On solid food surfaces, the progeny cells accumulate on top of each other, and in a couple of days they form colonies visible to the naked eye. A single cell may thus form a colony containing many millions of cells, which, except for rare mutations occurring during growth, are all genetically identical. The cells in nature and in laboratories, however, form a varied lot. Many variants are available with different metabolic characteristics. For example, a single small change in the genetic information has made these cells accumulate a red pigment and require adenine for growth. Besides vegetative reproduction, yeast can go through a sexual process. These 2 yeast cells are genetically different. Scientists usually work with haploid yeast strains. There are 2 mating types or sexes of haploid cells, called A and alpha. Separated from each other, they can be maintained indefinitely in culture. When 2 haploid cells of opposite mating type grow together, they exchange chemical signals which stop their budding, render them sticky, and change their form. The 2 haploid cells then fuse to form a single diploid cell, the zygote. Here is a mixture of haploid cells of opposite sex. They have formed clumps under the influence of sex-specific chemicals called pheromones, which are small peptides. Pairs of haploid cells of opposite sex fuse to form diploid zygotes, with a very peculiar dumbbell look. Many haploid cells do not fuse. Yeasts have no active way to look for partners. Now let's watch the conjugation of these three cell pairs. The pheromones stop the cell cycle at stage G1 and inhibit budding. Budding resumes some 2 hours after mating. The result is a mixture of